This is a Mission 5 flight experiment. It is the first oral presentation of the day from Fitchburg, Massachusetts. And when I announce the experiment, I will also announce the grade level, but remember that that was the grade level when they did the experiment last year. Actually, it was the grade level when they did the experiment. It might not even have been last year. So this is Fitchburg, Massachusetts, grade 12.
Good morning, we're the group from Montachusett Regional Technical School, otherwise known as Monty Tech, and we're Mission 5.
My name is Liza Anderson, and I was a principal investigator.
My name is Samantha Bracton. I was another principal investigator.
My name is Ashley Monroe, and I was a co-investigator.
My name is Stephanie Tivnan. I was also a co-investigator. And our teacher facilitator was Paula DiDiego.
Our proposal was that our experiment was to monitor the production of antibiotics from Bacillus Subtilis in microgravity compared to its production on Earth. And the antibiotic it produces is Subtilin, which is an antibiotic.
The way that our packaging was designed was so that we had one single tube with 2 clamps allowing for 3 compartments. In the first volume was our growth medium, in the center was the freeze-dried Bacillus Subtilis, and in the third volume was the growth inhibitor. 5 days before the experiments returned to Earth, we released clamp A and immediately replaced it, allowing for the growth medium to mix with the Bacillus Subtilis. And finally, 2 days before its return to Earth, we released clamp B, allowing half of the mixture to mix with the growth inhibitor, stopping the growth on one side.
We predicted that the freeze-dried Bacillus Subtilis could be activated and grown in microgravity. We also predicted that more Subtilin would be produced in microgravity. The analyses that we performed were an optical density test, a pellet mass test, a novel assay for Subtilin, and the beta-galactosidase assay.
An optical density test measures a material's ability to pass light. So the higher the number, the less cells present in the material, similar to how when you walk across the street, come across a storefront with windows, if there are posters or signs, the less light enters the building. So for our results, it would appear that since our experiment has a smaller number, there would be more cells produced. Using a centrifuge, we spun down our sample into both a pellet and a supernate. From this, we concluded that in microgravity, more Bacillus Subtilis cells were produced.
The purpose of running a novel assay test for the Subtilin was to measure the amount of Subtilin that has been excreted by the bacteria.
To run this assay, we took the supernate from the spun down pellet and plated it on a Petri dish. The Petri dish was coated in cells that turned blue in the presence of Subtilin. In our pictures here, that were taken a few days after we plated it, very few blue spots appear. However, by the end of our assay, it was clear that from this qualitative test, more Subtilin was produced on Earth than compared to microgravity, contrary to what we predicted.
Another test that we participated in was a beta-galactosidase assay. These graphs are the result of an optical density test comparing the gene expression of SPAS-LAC-Z and Subtilin. This occurs when exposing the genes to an enzyme. The first graph was a full-scale version of our test, and this graph is a scaled version of it. The green line, letter C, represents more production of beta-galactosidase and Subtilin compared to the experimental growth, which is the purple line, letter D.
The benefit of the knowledge gained was that we were able to learn that the ground truth produced more Subtilin antibiotic. We also learned that microgravity produced more Bacillus Subtilis. Future questions that we had would be, how would microgravity impact the production of bacitracin from Bacillus Subtilis? And we also wondered, would we see more production of Subtilin in a different strain of Bacillus Subtilis?
A thank you to our advisors.
Robin Gannison, who is a research associate at the UMass Medical Center in Worcester, Massachusetts.
And Dr. Abraham L. Sonnenschein, PhD from Tufts University School of Medicine in Boston, Massachusetts.
And a thank you to our sponsors, Montachusett Regional Vocational Technical School District,
Massachusetts Space Grant, MIT,
the Center of Advancement of Science and Space, CASIS,
National Center for Earth and Space Science Education,
NIPRO Foundation,
NIPRO Mold,
Leominster Credit Union.
Thank you.